in cells in the anterior pituitary. These cells produce hormones and release them into the bloodstream. Corticotropin acts on the adrenal glands located above each kidney. It stimulates the release of adrenal steroid hormones by the adrenal cortex. Thyrotropin acts on the thyroid gland in the neck. It stimulates the release of thyroid hormones. Follicle-stimulating hormone acts on the gonads. In females, it stimulates estrogen secretion and egg maturation. In males, it helps stimulate sperm production. Luteinizing hormone acts on the gonads. In females, it stimulates progesterone secretion and formation of the corpus luteum. In males, it stimulates testosterone secretion and sperm release. Prolactin acts on the mammary glands. It stimulates and sustains milk production.
Looks pretty complicated here, but it's really not that bad. Take it step by step. You have neural input, and you go through a release of hormones 1, 2, and 3, and the pink is just the way that the hormone is getting to the next destination. So with neural stimulation, you could get the hypothalamic releasing factors coming out. Those go into the portal system. The portal system delivers them to the anterior pituitary, and hormone 2 comes out. That goes into the systemic circulation, finds its target organ. In this case, if it's somewhere in the adrenal gland, then you're limited in what the hormone is, because the only thing that comes out of the adrenal gland the anterior pituitary that affects the adrenal gland is ACTH. And they call it corticotropin, but adrenal corticotropic hormones, corticotropin, that's the same thing. Then cortisol comes out, corticosterone comes out, finds its target in the peripheral tissues, and you have this ability to handle stress better. So that's your psychological effect, ability to handle stress better. How do you turn off ACTH or CRH? It's due to the level of the circulating cortisol and corticosterones. When they're elevated, then they suppress ACTH at the anterior pituitary. This, on paper, is a shorter loop than the one that goes to the hypothalamus. So it's referred to as short loop negative feedback. And this going to the hypothalamus is referred to as long loop negative feedback. The inhibition, then you turn off ACTH. Inhibition, you turn off CRH. If the levels are low, if cortisol levels are low, then you get stimulation by that decrease in hormone level of CRH release. Then ACTH concentrations go up, and then cortisol levels should go back up again. Then it's inhibitory feedback. So it's opposite of what the hormone in the periphery is doing. If the hormone is high, you inhibit the stimulating and the releasing hormones. If the hormone is low, then you stimulate the release of the releasing hormone and the stimulating hormone. So stress, just being physically or psychologically stressed, that's sufficient to get CRH released from the hypothalamus. Still goes into the portal system, whether it's due to neuronal or hormonal input. ACTH released from anterior pituitary goes to the adrenal gland, cortisol levels go up. This has effect on most cells. Your metabolic changes can be of benefit to handle stress. They also can be detrimental in that things like your immune system cells are suppressed in their activity. And also your cognitive levels under high stress, that changes as well. So you get a heightened alertness when you're under high cortisol levels, but if you're under high cortisol stimulation for long term, your brain adapts to that high level of cortisol. And even if the cortisol level just goes back to normal, all of a sudden your nervous system doesn't see that level of stimulation anymore, and it becomes depressed. You can have clinical depression just by reducing your stress level, which seems totally bizarre, but it happens. Because your brain is so used to that high cortisol concentration, and when it just goes back to normal levels, it's decreased enough that the brain is now not responsive, and you get bizarre thoughts, you get suicidal thoughts, and that can be very bad if you aren't able to recognize why that thought is there and that it's an abnormal thought and you really shouldn't be acting on it. A lot of people kill themselves when they have this depression because they just are responding to what's going on in their brain. This be better off dead, okay? If you're familiar with that football player who has the head trauma injury, what's his name? He was just recently on the news like last week. He's had so many concussions that he actually has this brain trauma syndrome, I forget what they call it. But he was saying that he has had all these suicidal thoughts. He says, but I'm too intelligent to act on them. But he says, unfortunately, some of my colleagues weren't. And so there have been guys who shot themselves and killed themselves because of this depression. So be forewarned, when you get out of medical school, graduate school, you may have a decrease in cortisol levels that give you a clinical depression. Do not act on those thoughts. Realize that the decrease in stress is something you need to do something about. Find some good stress to add back into your life, and then those symptoms will go away. Yeah.
And if you stimulate it enough that you keep yourself sane, does it like slowly go down the sensitivity?
Yeah. So the cells become less. They start pulling their receptors out of the membrane for cortisol. It's this sudden reduction. So you just submitted your dissertation to grad division, and like 2 days later you're graduating. That's a sudden reduction in stress. You've got everything completed, boom, your cortisol levels go down, receptors are still high, the cells don't see enough circulating hormone anymore, so their metabolic rate goes down, you get a clinical depression. If you resubmit some stresses, not that you're going to go and get another degree and raise that stress level up that high again, but just some kind of mental stress. For me, it was just going back to reading journal articles. And I had the physical stress because I was teaching exercise classes every single day, multiple hours teaching exercise. It wasn't physical, so I had to figure out what it was. Did I want another PhD? Heck no. So what could I do that had some component of PhD? Go to the medical library and start reading journal articles. That just snapped me right out of it. How many journal articles do I read per day now? Only the ones I have to. Obviously, you do adapt, and your cells get back to a normal response level again. Yeah, but in the meantime, that thought that goes through your mind, you have to recognize it as abnormal, what the source of it is, this sudden reduction in cortisol levels. Don't act on it.
Is that typical, about 2 days, then to take the plummet after the reduction in stress?
Yeah, it's probably pretty typical. It may take 40. It's not good. It's going to happen really quickly, though. Yeah. Depends on what else is going on in your life. If that was your major stressor and suddenly it's gone, then the crash happens faster. If you got other issues that are stressors, then maybe it will come down a little bit more slowly. But the thing is, recognize it as abnormal, don't act on it, but deal with it. Figure out some way of getting the stress level up till the receptor level naturally comes down. Okay, so we got TRH or thyrotropin, CRH, corticotropin. We have GnRH, gonadotropin, and GHRH, growth hormone, releasing hormone. They didn't do anything with that. The somatostatin is your growth hormone inhibiting hormone. So in that case, when you come back here, this negative, this negative here on the anterior pituitary is due to an inhibiting hormone, as well as negative feedback. And prolactin, prolactin has inhibiting hormone. Turns out it was dopamine, which is just another catecholamine. So summary over here of what it does: thyrotropin stimulates TSH or thyrotropin being released. ACTH, corticotropin is being released. Gonadotropins, LH and FSH are being released. Growth hormone. And interestingly, somatostatin is also decreasing the growth hormone, but TSH release as well. So double inhibition by somatostatin. And that's it. Okay, so growth depends on growth hormone being there, somatomedins being there. Growth hormone alone, not good enough, got to have somatomedins. So the hormones, those two, one being permissive of the other, has to be there. But then there's a lot of things that are also involved. What do you inherit genetically? If you are genetically going to be 5'6", there's nothing that your diet and hormones are going to do to get you to be 6'5". And they have tried with people who had pituitary insufficiency, growth hormone insufficiency, try to supplement the growth hormone. And when they tried to give too much, it had disastrous effects. And they found that the only return with proper supplementation of growth hormone was maybe an inch or two of height. So somebody who is growth hormone deficient and should be 5'10" or 5'6", but is 4'6", you're only going to get them to 4'7" or 4'8". If you try and push them beyond that, then the whole system is going to unravel and you're going to have a really sick individual on your hands. So genetics, you're predetermined, but hormones can influence what your genetics are. Diet in Japan during the wartime and actually pre-war, the Japanese very short of stature, but a lot of it was due to lack of calcium in their diet. They had bow legs. Once they got the calcium and the bones straightened out, they were less, had less osteomalacia. Bones were straighter. They got better height. And then when they got better nutrition all the way around with the American diet of the 1940s, not the current American diet, but the 1940s diet, which had better all-around nutritional value, then they started reaching their maximal height. So they went from low 5'2", 4" range up to 5'8", 5'9", 5'10". So they gained quite a bit of height. But now they're gaining quite a bit of bulk because they're too into the high-fat Western diet, and they need to get rid of that like the American and other Western diets are trying to do. If you have chronic diseases, that's going to influence what you can get in the way of nutrients. Maybe what you can eat, what you can digest, what you can absorb, what you can utilize. So anywhere around that level. And then the diseases themselves might be attacking muscles, bones, and connective tissues. So if you have a chronic disease early, you may have severe stunting of your growth. The cystic fibrosis, where it affects the ability for oxygen to get in and out of the lungs because of all that mucus, and also the pancreatic enzymes getting from the pancreas to the digestive system to digest food. They have like a double whammy. They don't have the oxygen for ATP production, they don't have the enzymes in the duodenum to digest their food, so they don't have the nutrients to grow. So people with cystic fibrosis usually have severely stunted growth. You have two major growth spurts. The ones just after birth. So the baby comes out maybe 6-8 pounds, and then by the first year, it may have tripled, quadrupled its weight, and then the next couple years, it's growing really fast. Then the growth kind of slows down and then takes another spurt at around 12. Depends on when puberty hits, but usually puberty is around 12. It's getting earlier and earlier now. Then you have a second growth spurt. Growth hormone is, as I said many times already, is essential for growth, but without somatomedins, you're out of luck. So liver problems, no somatomedins, you don't have proper growth. The growth hormone, gotta have IGF or somatomedin. It's going to stimulate protein synthesis, collagen, so ligaments, tendons, all connective tissue has some collagen component. So that protein, if you don't have growth hormone, that's going to be insufficient. Also, bone development. What you lay the calcium and phosphates down upon is your collagen that is arranged spirally in the bone. So again, you need that protein synthesis. Also, growth hormone also influences fatty acid levels in your blood. And the growth hormone actually will mobilize fats and increase fatty acid content, and then spare the blood glucose for brain use. So your cells and tissues will be going into beta oxidation for ATP production. So the growth hormone the glucose can be used by the brain. There's a diurnal rhythm, like the cortisol, high and low, high and low. So 2 phases of growth hormone release. 1 hour after sleep, that's when growth hormone is the highest. And then it will start to come down through the rest of the night and into the day. Growth hormone release is also stimulated with heavy levels of exercise. So the muscle bodybuilder guys, they want to get their maximum sleep possible, and they also want to have their maximum workout possible to get maximum growth hormone release, hopefully without growth hormone supplementation. So exercise will stimulate the release of growth hormone. The stress levels also do, but stress has its own negative effects on muscle and connective tissue. So you don't want too much stress. Blood glucose levels going down, that will promote growth hormone release. So you'll find these bodybuilders also eating a very high protein diet and trying to stay away from carbohydrates so that they have a lower blood glucose concentration. The increase in blood amino acids, decrease in blood fatty acids, those are, and then ghrelin from the stomach, those will have stimulatory effects on the release of growth hormone. So the growth hormone, again, to the liver, and it's going to have overall metabolic effects on tissues like adipose muscle, liver, metabolism, and then bone growth as well. So a little bit more about your biological clock and its control. There's a nucleus right above where the two optic nerves cross. That's your chiasma. So your optic chiasma. That has, right above that, the hypothalamus has a nucleus called the suprachiasmatic nucleus. So it's named for its location. This collection of cell bodies in the hypothalamus are responsible for setting your biological clock. So when you wake, when you go to sleep at night. But it's influenced by the light-dark cycle. So each day it's going to, when the sun comes up, when the sun goes down, it's going to influence the stimulation of these neurons, cell bodies in the suprachiasmatic nucleus. Okay. Somewhere along in here, there's, next chapter, there'll be more, I think, about how your eyes and your pineal gland will get hooked up. Or, no, I guess it's coming up. Okay, so pineal gland, when you get the pineal gland stimulated, the melatonin is released. And in the dark, melatonin goes up, in the light, melatonin goes down. So dark stimulates or inhibits what are called intrinsically photosensitive ganglion cells. So these ganglion cells here, they're intrinsically, meaning they respond to light themselves. Majority of your ganglion cells are responding to what the retina is, the retinal receptors, the rods in the cones are telling them via bipolar neuron communication. But you have some intrinsically photosensitive ganglion cells that will fire first. So if you look at the retina, the light's coming in this way, your retina's back here. You got these big ganglion cells that the light has to go through in order to hit the rods and the cones that are in the retina. The photosensitive ganglion cells will fire due to the light coming in and hitting them. So they'll start sending action potentials to the brain even before the light that's going past them gets to the photoreceptors. So they're sending information to your brain, and before your brain actually knows that the light is bright, guess what happens? Well, before you squint, pupil constricts. So your pupil constricts in response to the bright light, not because the retinal receptors are telling you the light is too bright, but because these photosensitive ganglion cells have fired first. By the time your brain is aware of the light being too bright and you're squinting, then you already have your pupils constricting to help protect the retina from that bright light. So you have the ganglion cells sending information to this suprachiasmatic nucleus, your master biological clock. What that does is stimulate production of your clock proteins, and those clock proteins then synchronize your light-dark cycle. The degradation the rate at which they're broken down, then can re-stimulate the suprachiasmatic nucleus to release more clock proteins. So if this degradation rate goes up for some reason, then you'll have more clock proteins being made. But if the synthesis is adequate, then there will be negative feedback to the master gland to turn off that stimulatory effect. So melatonin, where does melatonin come in? Well, that suprachiasmatic nucleus, while it's synchronizing these clock proteins, it's also sending stimulation from the hypothalamus back to the pineal gland. The pineal gland will then start releasing melatonin in the dark when this is inhibited, which means this is not stimulated. Then the suprachiasmatic nucleus not stimulating the pineal gland, not inhibiting the pineal gland, melatonin production goes up. In the light, the increase in the stimulation of your suprachiasmatic nucleus will inhibit the pineal gland, and melatonin production goes down. So the increase in melatonin, then you're going to, that will skew your light-dark cycle to the resting, relaxing side, the sleeping side. The light will decrease the melatonin production, so you have greater awareness during the day. You have greater mental acuity. People who pop melatonin pills to go to sleep at night, they have to take some kind of stimulant during the day to make them function better during the day, otherwise they're going to be groggy. So they're popping pills to go to sleep, they're popping pills to try and wake up. Depends on what the pill they're popping to wake up. If that's an amphetamine, then they got their own problems going on there with that too. But if it's caffeine, then they can have caffeine jitters. You get too jittery with your stimulant, then you have other problems.
Question.
Yes.
Can the suprachiasmatic nucleus be stimulated via light to the skin? It's a
No. It has to be light to the eye. So the optic nerve will carry information. We'll cover this when we get to special senses. But it has to go actually to the midbrain, and then it has to come forward, and then it has to go down, it has to come back up to get to the suprachiasmatic nucleus, and then back to the pineal gland.
That's the only way to change the circadian rhythm.
Is via the light and dark
I was reading a
cycles.
study of a guy put flashlights in the back of his legs when he was on a plane in order to get his circadian rhythm going. So he wouldn't get jet lag off the plane. And his study showed that it was working.
Well, if he's looking at the light, yeah. The incandescent light will work.
It was in the bottom of the knee.
Huh?
Both legs. It wasn't looking at
Yeah.
As far as your brain is concerned, if it's like a LED or
That's the one.
fluorescent light, you're in the dark.
I didn't finish up with her.
So I
then Glenn
suppose
told
it
me to jump on
depends
pickup.
on what kind of light
And I
is
didn't
on
refill
the
her other
airplane.
things because it was
So
like some kind of
if
insurance
it
thing.
is incandescent light on the airplane, that would screw up your light-dark
He
cycle.
grabbed his own flashlight.
No, but what I'm saying is, if the airplane lights are suppressing your melatonin production, that might lead to more jet lag, but probably not significantly. So I don't know if this case study will hold up. I will wait and see about that.
Does she still do
No,
eyelash?
Yeah, that's not the same size.
Does she have like clients?
Interior lateral ligament.
Oh, you want to do the all ligament?
She makes so much money off of it.
Does she?
You haven't
Yeah,
told me
because
about
like
it.
I remember I did it one time.
Yeah,
It was
but
like,
I was just gonna ask you about
Where'd
it.
you go?
it was
Okay.
like somewhere
Sit
in
down.
Milwaukee. It was like 25, 30, I think.
Are you serious?
Yeah,
pretty
I got it cheap because like it was like First time members get half off. That's why I tried. It's supposed to be like
Yeah,
50.
he's stalling
That's what she
because
charged.
I think I could be
That's still pretty cheap. It's like,
Is it?
yeah.
okay, so peripheral
I'll tell you later.
hormones, if they're hydrophilic, they must have a receptor in order to have a message sent to the inside of the cell. So this is one of the ligand types of receptors that are in your plasma membrane. It's got these
one of the side.
1, 2, 3, 4, 5, 6, 7 loops back and forth through the plasma membrane. That will ultimately be linked to a G protein. The G protein will be linked to an amplifier enzyme. The amplifier enzyme will be linked to a second messenger system. By the time you get to the second messenger system, you have one hormone that now has activated via the G protein several amplifier enzymes, and each of those has recruited this second messenger system to get us another level of amplification. Each cyclic AMP, in this case, can only activate one protein kinase, but each protein kinase can phosphorylate many enzymes or proteins, and then those can have multiple effects inside the cell. So by the time you're finished, you could have one hormone now suddenly amplified 10,000 to a million, 100,000 to a million times inside a cell. So although it seems inefficient to have something stuck on the outside, it actually is highly efficient in stimulating with this amplification internally of the cell response. Is highly efficient in getting effects in the cell. This, I don't know why when I scanned it in and cut off so much in the presentation mode. But up here you've got your amplifier enzyme, and this is creating a second messenger, but here it's IP3, so inositol triphosphate. This is linked to a G protein that stimulates phospholipase C. What you don't see is that you have another second messenger linked to phospholipase C. It's in your textbook though, you can see it there, is diacylglycerol. And that's lipophilic, so it stays in the plasma membrane and then activates protein kinase C. Inositol triphosphate tends to open calcium channels. Could be in the ER, it could be calcium channel in the membrane, but overall calcium then is increased intracellularly. The calcium itself, you might have calcium channels that are being opened by your G proteins. And calcium can be its own second messenger. So IP3 releasing calcium from the ER, the calcium can act as a second messenger in addition to IP3. Calcium coming in from the extracellular fluid also can stimulate release of calcium from the ER that we saw in the heart. Okay, calcium-induced calcium release in the heart muscle, and then also from the scant endoplasmic reticulum in smooth muscle, calcium-induced calcium release. So here we have calmodulin being activated. We saw that in the smooth muscle of the physiology of the muscle, and that activates the light chain kinase, and that will then phosphorylate downstream your myosin light chain. Okay. Thyroid. So thyroid gland, so here we've got pretty typical thyroid follicle cells. They're cuboidal in shape. The cell has its nucleus centrally located. the cytoplasm equally distributed around it. The little lighter areas, that's a sign that the colloid has been endocytosed, and that means the cells are checking it for iodination. If there's a 3 or 4 iodines on the tyrosine, then it will be released into the blood capillaries outside the follicle. If there's only 1 or 2 on the tyrosines, then everything will be recycled back into the colloid. When you look at the follicle here, it looks like this follicle is huge, this follicle is small. But you have to remember the thyroid is a gland of multiple follicles. When you take a thin section through the gland, you're only going to be cutting you're not going to cut all follicles in the middle or all follicles at one end. You're going to catch some at the top, some in the middle, and somewhere in between. So the size of the lumen is determined by where you cut that particular follicle. All of these follicles have signs of reabsorption. All of the follicles have cuboidal cells. So probably normal circulating T3s and T4s, which turn off when they're elevated, turn off TRH, turn off TSH. And so you don't get overproduction of colloid. You don't get over reabsorption and release of T3 and T4 either. If you did, then all the follicles would have very little colloid in them. They have much more lacunae, these little resorptive open areas. All the follicles, the colloid would be small, and you look at the cells, they would be very tall because they're overstimulated to produce T3 and T4. Calcitonin secreting cells, these C cells, they appear as very light staining cytoplasm with a nice light staining nucleus in the center. And that's very different from these darker nuclei of the cuboidal follicular cells. So you need tyrosine and you need iodide in your diet. So iodized salt, go to Long's or Safeway and look at the salt. One will say Morton salt, iodized, Morton salt, plain, or generic salt, iodized and plain. It's the best source of iodine. Don't need to consume a lot of that salt, just a little bit. Boil your pasta in it. You sprinkle a little of that on your food occasionally. You have adequate iodine for your thyroid hormone. If you eat certain vegetables, you get iodine. So if you're iodine deficient, it's probably because you're not getting it in your diet. You can also become iodine deficient if, I think I mentioned this in Martini's chapter, if you OD on mustard or you OD on yellow turnips, that will interfere with the conversion of iodide to iodine. And if you come in contact with propylthiouracil, which is used to treat hyperthyroid conditions, but oftentimes is in your basic lab in rich, just basic labs that you're working in, might have PTU, and if you come in contact with it, could send you into a hypothyroid condition. The thyroid follicle cells also have to make this thyroglobulin. That's what is put out in here that forms that amorphous gelatinous mass called colloid. That's what your tyrosines are attached to while they're being iodinated. So a lot of amino acids have to be taken up to make that protein. Okay, so if you have one iodine attached, you have a MIT, a monoiodinated thyrine. If you have two iodines attached, you have a DIT, a diiodinated thyrine. You have three triiodinated thyrine, and then if you have four, it's tetraiodinated thyrine. This one is what they call thyroxine. T3 is your metabolically active form of thyroid hormone. Your tissues will convert T3 to T4, so if your tissues really don't care. As long as they're getting T4, they can convert it to T3. They're getting T3, they're able to use it right away. Okay, so next week, well, we do have lecture Monday, Wednesday, so we'll pick up on Monday with what's going on in the follicular cell.